Hello and welcome everyone to the 23rd webinar in Contact's Produce Safety Webinar Series. This third season continues to tackle fresh produce safety topics through a dynamic webinar series that brings together industry, academia, and regulatory minds to solve some of the produce industry's most timely challenges. This series is part of a larger industry outreach and risk mitigation project called Contact, or Scientific Challenges and Cost-Effective Management of Risks Associated with Implementation of Produce Safety Regulations. This project is supported by the Specialty Crops Research Initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of the speakers and do not necessarily reflect the view of the U.S. Department of Agriculture. So who is Contact? In total, we have over 50 faculty, staff, students, and volunteers that make up Contact, not to mention the support of our Produce Advisory Committee, or PACT, which we rely on heavily. It takes a village, and we appreciate each individual's support and ongoing efforts to make Contact a grant championed by you all. We've been featuring previous Contact research, but we wanted to bring attention to other USDA SCRI proposals as well and grants. This one is a recently published paper from Dr. Martin Weidman's SCRI, but this one features work from Virginia Tech, the University of Florida, University of Rochester, and Washington State University. Here, the project goal, we wanted to compile a very large data set. This project happened during the COVID-19 pandemic of various regions, water sources, and methods. And with this data, we wanted to look at assessing methodological differences and how they affected observed microbial water quality, and then to determine if those methodological factors influencing the observed microbial water quality could be distinguished from other non-methodological factors such as water type, weather, and land use. This might be a pretty cool paper since the FSMA Produce Safety Rule is relying a lot on agricultural assessments of water. And here you can look at pretty good coverage throughout the United States, and also picked out key factors to look at for Salmonella, pathogenic E. coli, Listeria species, and also Listeria monocytogenes. One of the really nice things also in this paper is that there's a nice table of best practices to use when it comes to tidy data, looking at water quality data, or even collecting and organizing your own water quality data that you can use for trend analysis and future efforts like an ag water assessment. Be sure also to look at the other Contact resources. We have a really great website and YouTube page that include things like our key takeaways, our training courses, as well as our webinar recordings and so much more. You can subscribe to our page to get instant notifications of our content as well. Trevor does hold the faculty title of Extension Research Specialist Emeritus at the University of California, Davis. With his wife, Trevor recently founded T&K Suslow Consulting. He was also supposed to be retired, but he's clearly not doing a very good job of being retired. But for all of you that don't know Trevor, he's definitely Mr. Produce, so the floor is yours.
Lessons and other tall tales from the field and rapid response support. This can be a real challenge, and that is actually one of the things that maybe we can explore at the end. There is an understandable factor in really going into, and sometimes there's a regulatory barrier, there is a litigation barrier, and I'm certainly encumbered by a lot of that, but I'll do the best I can to give some of the examples of takeaways. And if you go back, if I'm assuming it's still available, look at the first webinar I did, very broad, addressing various risk factors. So I'm going to jump on in, and this really arises from a variety of experiences and a level of frustration, if you will, for what I've always dedicated in my career to, in being that extension specialist, being that bridge between that very essential and important continuum from fundamental research through more applied research to extension and outreach and both directions, bringing issues and needs for problem solutions from the outside world, whether it's industry, environmental, public health, whatever it may be, to research and the opposite direction. But what we see more and more, there's this rising challenge for translational adoption and rational and sensible produce safety metrics based on science development for how the drive for socialization of research outcomes through press releases and institutional blogs, always pushing for impact. I get it. I understand it. But when you read that, my interpretation, and again, fortunately, she provided you that disclaimer at the beginning that these are the opinions of the speaker, not USDA or necessarily members of the contact group. I would argue that they are quite often exaggerated. Quite often, that's not necessarily the responsibility of the research team or the PI, but more their media person. I have had to deal with this myself, my own institution. So either exaggerated or unintentionally biased. And if you're only reading that, which many people do, or if you're only reading the abstract summary, you're going to get one message, and it may not be one that I always put in the column of, again, I'm speaking to industry here, goodness of fit. These are widely republished in social and news communications in general. Most, I would argue, this is a different crowd, right? But most never read the journal paper. And there are some real challenges for them to read it, understand it, and appreciate it, or they read the beginning and the end. And there's a lot of barriers to understanding the methodology and how the methodologies support the conclusions. But quite often, each time there is an extreme industry or an agency or some industry group reaction to it, and may be very appropriate, it may be misled by not understanding what's really in the paper as it relates to the outcomes. One of the sort of leading, we always call rumbles, that I've tried to make rumble throughout this presentation to the end, is that continuing to work on coordination and communication are key, but I've always been a very big proponent of appreciation of the diverse cultures involved in a lot of this, and mutual awareness and understanding of the cultures and the sort of what drives each one, and the language and understanding across industry, across agencies, public health or otherwise, academia, and I put academic extension into a slightly related but slightly different category, generally will lead to better design, better objectives, better use of funding, and better translational research adoption. So quite often, as we often say, I'm sure many of you heard, the research, they hit every deliverable, but they didn't hit this. They didn't meet the so what barrier. So where am I heading today with all of this? I'm going to try and give an example where, largely from the research standpoint, and certainly what one sees today as a reviewer of research proposals, or sitting on panels, trying to read, but also retrospectively looking at the papers, that an understanding of supply chain structure can really help with research design, and therefore ensure a better acceptance and acknowledgement and use of all of that effort. So then move to some high-level examples from field responses. I have to put the brakes on, remembering that this is going to be recorded. Again, there's a lot of sensitivity around this, but I'll do the best I can to at least share some key learnings from a variety of tremendous, unbelievable field experiences I've had, leaning a little bit more on more recent, but also some from the past. Walk through an example of a root cause assessment. Fun to do that. Again, real world, real-time response. And then suggest a rapid response capacity building opportunity that I'm hoping will lead to some interaction and comment from the audience, or at least questions. And then hopefully there's a point to all this today, and where I hope to end up is a sort of a point of encouragement for the future. So many of you are going to be well aware of this, but I think it's clear, at least it seems clear to me from interacting with a lot of young researchers, even some well-established mid-career, and reading grants and reading some papers, that there isn't a great appreciation for how the outcome of objectives and proposals would be viewed by industry based on their structure, based on the sensitivities of where that may lead either intended or unintended consequences. Just walking through this without going in great detail, you have what we say a grower, farmer, same thing. At a ranch, farm, same thing. So there are many times when, the grower is also the shipper. We'll come back to each phase. There may be multiple harvesters in the same field because you're growing, selling to different customers. Then you may have a contract harvester. You may be going to a specific sister or shipper or money shippers. You might also be the shipper yourself. You might also be the processor yourself. There are many processors. And you may be co-pack arrangements with other processors. And then ultimately you have customers and consumers. Again, just a very sketchy overview of the complexity of the supply chain, particularly complex for leafy greens, but true for many other commodities as well. When you're thinking about research or when you're trying to go in and understand with problem solving or a root cause assessment, I tend to stay away from root cause analysis. I'll probably share that with you a little bit later, if I assume I get there, that it's almost never linear. So who owns the land? It's not a farmer or grower sitting on a tractor. Obviously, there's that too. But we tend to deal with quite a bit more of this. Who owns the crop? It's not always straightforward. Who manages the crop? Who harvests the crop? Who cools the crop? Who processes? Under what label? Under what brand? Or multiple labels? Who arranges for the transport? Who consolidates and distributes? And who is the end customer or consumer? All of these things can influence how a research proposal, research objectives, and certainly research outcomes and setting of standards, policies, and best practices are received. So again, just very briefly, the grower at the ranch usually runs the farming business, not generally driving the tractor. You may or may not own the ground, which has some strong implications, particularly as it relates to food safety. There are some other with soil-borne pathogens, things that impact the value of the ground or its use. And typically they're going to farm multiple locations. They may rotate ground with other growers. So again, the kinds of things that are possible when you see outcomes saying they should move to a different this, or they should start doing a different that. Within the structure of how that ground is used or leased, long-term leases, that may or may not be practical or possible economically or for various other reasons. Many grow something on the side. This also interfaces with traceability, which I'm not really going to talk about. But basically, the growers is driving it that way. And particularly from this example, again, drawn leafy greens, which has received a lot of research attention and that continues, sustainability, a different kind of sustainability, profitability, really requires some pretty quick turns from crop to crop. So you go from crop to harvest to disc it down to replant. And there are emerging and pretty strong indicators for relationships to seasonality as it relates to contamination, as it relates to understanding the patterns of outbreaks that have occurred or patterns of positivity in pre-harvest tissue testing. Again, I won't be going into details. I did deal with it a bit more in the first webinar that I did for the Contact series, but there's a lot of greater understandings. And what's so amazing for somebody like myself is the tools to explore this, the tools to really query this and explain this are so tremendous today versus what we would have had when we were beginning. The questions are the same, but the tools to resolve those questions have improved tremendously. And the young scientists, those using those tools and becoming skilled at them, is really going to make a huge difference. Shifting to the harvester, just again, driving an understanding of what's involved and how the structure works. The shipper may have their own harvest crews. They may use custom harvesters. The grower may have the responsibility. The processor may have their own crews, or they may use custom crews. There's just, again, so much diversity in all of this. And again, one thing to understand in terms of how one might approach what I would say the most usable, the most solution-directed research is to understand when that harvest happens, how that relates to research you're setting up. And as Laura mentioned, in just tell me what to do exactly these days probably won't cut it across the board particularly as it relates to agricultural water quality assessments. Harvest crews work well before or shortly after daybreak, and baby leaf harvester may happen even earlier, particularly during the hot weather. The shipper can pack, cool, and sell the product, often contracts with the grower to be sold under the shipper's labels, and generally arranges for harvesting, generally owns or participates in the cooler and shipping operation and contracts with multiple growers. And then a different type of receiver would be a processor. They're both handlers, but they would do the things you're familiar with now, make packaged salads, value-added types of facilities. They may have multiple facilities right close to the source of production or shipped to regional locations or everything in between. Typically, they have the large staff of food safety and quality control specialists, and this is where a lot of those food safety professionals work, and they provide a lot of the guidance, or they can. The customer base can vary a lot from hyper-local all the way to more international. And of course, I'm not going to read through all of these bullets. I left it in that may be an opportunity to look at it if you're not familiar, or just again, where does all this go? The customer, not the consumer, really the customer, a variety of different types, a variety of different ways it enters those markets. And the same product can go multiple directions. Again, there are some implications, again, for how practices, how any type of change in what's done, but also what drives the industry, which is very dynamic. And working with outdated and old-fashioned paradigms is not going to support an effective research strategy and objective. So I'm going to shift a little bit into things that relate to these sort of tall tales from the field that Laura asked about, and some of these experiences that certainly help frame my understanding of some of the challenges, and always try to turn those into research opportunities for myself, for others. And you'll hear me probably say Center for Produce Safety several times throughout that, but also within the USDA NIFA program. So one that I've been concerned about for a while, I don't have a really dominant patience gene. I fully acknowledge that, but letting things evolve over periods of time when you know that there's science, you know that there's data out there to argue for a change is tough. But the risk associated with surface drip, I think, has largely been overlooked or pushed to the side. This happened to be a rapid response effort due to contamination of celery with E. coli 15787. Again, you don't always get to an absolute causal agent, but certainly some strong presumptive explanatory things where the drip looks a lot more like a furrow irrigation from a water quality because it was drip, not receiving the same attention as an overhead irrigation. Yet I'm just trying to gloss over examples from actual events, but a lot of then contact that can be transferred. You'll hear me say this a few times from soil. So I do say contaminated water can create contaminated soil, and it's very different when you do pre-harvest tissue testing and sampling from what harvest crews experience. And just in case you're not aware, that's very different from a subsurface drip application, although I hope by now most people are familiar with this, or even subsurface with the protection of some sort of row cover mold. In any case, so again, just pointing towards things that I think continue to be areas where additional risk research could probably help refine the standards and metrics of industry if they're open to receive that information. Related, sort of continuing along on the theme of agricultural water, because this is where a lot of people's experiences will drive from seasonal canal disturbances. And just picking one example, high wind, I think you can see the movement of the water in those canal system agitates sediments. We know that's where You have, you can't have pathogen residency. Multiple testing that I've done in the past, and I think that's shared by others, is that in general, the types of filtration used if you're going to be doing drip do not generally restrict the passage of bacteria, depending on what's going on. And some of that is different clays or silt or sand, but bacteria will go through, and we've done that many times on both sides of the filter. It's really not that different. So in this particular case, there were high E. coli counts, just generic E. coli encountered on snap peas, but that was a cause for rejection. It's a multiple harvested crop. It was all on surface drips. The grower was very puzzled as to why that was happening. But again, harvest of this particular crop by large crews, flipping the snap peas back and forth through multiple harvests creates additional soil surface contact. So again, that was a pretty plausible explanation. Didn't go in to look for pathogens, but ended up shifting some other practices that help protect the water source from these fecal indicator intrusion. Put this one on. I'm not going to be sharing a lot of data, but it was fun to pull this out from the past where we're trying to set up to be able to understand die-off. This is one of the very early attempts at comparing, looking at a naturally occurring, very prevalent organism, Citrobacter, different ones compared to some other. So within growth chamber, and what is the effect of an acute desiccation? So again, with a crop like parsley and cilantro, which was problematic at the time, what one might expect from die-off. That this is the first indication that, of course, there's going to be differences. This has been well established multiple times. Now, same sort of issue, though, from a crop basis, where contaminated soil can lead to contaminated product during harvest, and that's very different than what you may find if you're just assessing die-off of the crop itself. This is an aside. I threw it in just because these are great undergraduate student projects. There's clearly, if you have a really compromised, whether it's furrow, drip, or otherwise, systemic transport from roots to shoots of cilantro under controlled conditions. We set up different trials in the field with surrogates, at least under those conditions, did not observe that to be an issue. The same thing with multiple cuts of something like cilantro. So clearly, outbreaks have happened. Clearly, there's a timing involved in all of this. But one of the points here is that as there is this expectation to really try and delve into the data that's available on the persistence of pathogens post-irrigation, particularly overhead, I think we underestimate the survival capacity for these things under different weather conditions, and expecting a grower to be able to sort that out on their own certainly is not what any of us that are involved in extension, outreach, and support, and certainly not those that are very much involved in doing the more fundamental studies as it relates to the kinetics of survival and persistence and recovery. So just another example from the field studies that were done with cilantro at the time. Not going to be going into all the different facets of that, but just making a couple of points. If you believe that within the sampling that was done, pretty well replicated observation that in general, Salmonella will persist longer, survive better under the same conditions than 015787. But the real point here is that 12 days out, Davis under spring conditions, but still pretty warm. If you believe this 1 out of 15 samples, that was the level, resources and staff power at the time. Think of the size of the field that I just showed. Think of what that means in terms of the populations of plants, if assuming that's real. Obviously, we're inoculating it. So even at log 4 per mil, and then you have 12 days of die-off, you always seem to have these persistent populations that have found protected sites. So that's really the point is, again, there's a really limited amount of commodity-specific, condition-specific related to practice specific, including some work that I'm not presenting on how agricultural chemical applications impact survival and persistence generally in the positive way for the pathogens, or at least their surrogates. All of those research questions remain. And the beauty is that there are a lot better tools, a lot better understanding of how to go about that from a statistical standpoint. So we're going to hop around a bit. Hopefully this doesn't make everyone too crazy, but this interface of animal and specialty crop production has triggered several rapid response support events. A number of them have been on particularly cantaloupes, but some other specialty melons, and still having to deal with this in not in the mainstream with the most aware firms, the most dedicated firms to this, whether California or elsewhere, and have been across the U.S. and into Central America and Mexico dealing with some of these issues. You more often than not can point a pretty strong finger back at water. And again, water treatment, the understanding, the research done within Contact, within the Center for Produce Safety supported research, and other USDA supported research and state supported research is really helping to define the database from which I think the industry can really adapt and adopt, I think with some help from a variety of sources to really understand how that information, how that data fits into their specific programs. Another example, just to spreading the joy, dairy lagoon waste released over many years consistently to adjacent tree nut orchard, specific sort of extreme weather event, changed that risk profile, that risk balance. And in this case, runoff impacted adjacent specialty crop farms. Was that dairy lagoon waste released to an almond orchard a good practice? You could argue a variety of, depending on how that soil die-off might occur if there's something in that water. But again, it's all driving at that understanding adjacent land uses and nearby land uses, and how that can impact both your water, but also the farm itself. This one, I took out the information, but basically this is not, it's a rare but not a unique event where you have a wastewater treatment facility near a specialty crop production. In this case, this one happened to be walnuts, but leafy greens, strawberries, melons, a variety of things. And that's not a surprise, it's there. But in some cases where that buried black water sewer line is, can be a surprise. And so when that happens with a perennial crop, a huge question, what do you do? Who's responsible? I'll leave that out of the discussion, but all of these things should be ideally captured in food safety risk assessment. In this case, it was a surprise for the grower. So I'm going to continue on this theme of just sharing with you some things related to assessments of changing and being aware of adjacent and nearby hazards. And for some of you that have taken some of the early advanced good agricultural practices courses we held at UC Davis, you may recognize this, but I'm hopeful that there's a lot of new folks in the audience because time does march on. So I'm going to go through this. Normally, this would be a breakout group challenge example to explain, but I'll walk through it. And again, this is altered a little bit to try and protect those that were involved, but it's pretty true to what happened. A pre-harvest study of the prevalence of fecal coliforms and E. coli on fruit and two large ranch blocks, growing apples blocks, about 3 miles apart on the river course, water drawn from the river. The results were very low levels of fecal coliform and E. coli. And given those results, no further testing was done. You can see the characteristics. There's a town nearby. Year one, when this started, they were about that size. It started to expand. There were some other seasonal row crops involved. Over time, things changed and they weren't really being tracked. And a major customer required microbiological testing of all lots of fruit within 7 days of harvest, which was the theme at the time that was being pervasive throughout the industry. And this time, an average of log almost 6 fecal coliform per fruit and log 4 fecal coliform per fruit for the block A, and somewhat lower, but still in the detectable range for Block B. So what kinds of things changed that weren't really being thought of as being potentially a risk factor or hazard for the grower? A great expansion of the town. Because of the expansion, there was a regional park developed from sort of year +3 with a lot of activities, including the public access trail with a lot of dog, whether that was related or not. Never explored that, but you have a lot of free running dog. And then the development of ranchettes, phase 1 and phase 2 on both sides of the river, and the sort of added information I left out. They were zoned and allowed for chickens and for goats and for small animal corrals and a variety of things. So again, the point here is just paying attention continually to things that could affect your water, could affect your farming operation due to stuff that is beyond your control. So that was just an example. And then this one, I have to be careful as always, but this really played itself out potentially in some respects where the rural neighbor sort of historical visitor status was often overlooked. And you just happened to be there at the time to observe horses and horse droppings and dogs and other things involved. And there's another layer on this, which I'll come back to later in a couple of different events where even though the potential contamination could be very small from these potential sources, you add in a amplification factor with a unusual or extreme weather event, and now you have a recipe for, I believe, what at least impacted that crop, whether it was directly involved in an outbreak or not. I'll leave it at that. And shifting again, some of this is going, but sticking with the weather-related theme here, the tipping point for a risk shift and root cause is not always apparent by the time you get there. And I'm sure most of you have heard this many times. Certainly, this is a real challenge for FDA, other investigators involved, as well as those that get pulled in to assist with outbreak response, environmental investigation response. If you look at this sort of product handling conveyors, this is at receiving, you can look at that. It's not well maintained. It's got a lot of impacted mud. It can assure you, having been there more than once, it doesn't always look like this, but when this is done as a challenge question for training purposes, would your opinion of the level of risk change if you knew that this receiving line was outside? And at this particular point, at a particular time, which caused the observations, which caused the detection of concern or may have contributed in some way to an outbreak in one specific case, impacted by multiple days of rainfall. So now you've got an outside receiving line meant to typically stay dry that became impacted by unusual seasonal rain. So again, digging into the history, digging into those facts is part of the process, but when you walk up, it's not always obvious. But that has happened more than once in various things I've been involved with. I put this one in for fun, just to make a point that sometimes this kind of work, this is an encouragement piece for those that are in postgraduate training or interested in different aspects. That I just love applied research. I love trying to solve problems. I'm not doing a lot of acknowledgments here. Obviously, I don't do this all by myself. Tremendous industry partners, academic partners, state partners, but working with FDA and other regulatory agencies. But here, it's just George Nikolic was just a tremendous asset. Through our contacts with the Center for Produce Safety, of course, I had known him many years, trying to resolve produce safety with brush design, deployment, and various post-harvest water use efficiencies. It was a great, fun project, but there was a handoff needed. And this is often true, that we got started, looked at a variety of things. He designed a really great pilot system, and there was a handoff when I stepped away, went to Meijun Zhu up in Washington State. I'm really excited to hear what she has to report this year at the Center for Produce Symposium. What was learned really held no surprises. And this, again, is how we look at research, or just encouragement from a lot of effort, true for many different things, not restricted to fruit. But in a pilot system, removal from fruit is inconsistent. No big surprise, lots of research to support that. Dose is more important than the number of spray lines. A lot of other things that George and I started on, and then hopefully Meijun and George finished. But the other one in red is really the key point I wanted to make here. That validation of risk mitigation requires very large numbers of fruit. And you really have to debate and think about how you're going to do the challenge inoculation between full fruit immersion. I'm not a fan of, I'll just state that and I'll let it go there, to spot inoculation. Both has pluses and minuses. But when you're operating at that level, I think it explains a bit more the kind of exposure that consumers would have, where you have large numbers of fruit, all with the same, but only a certain number of them end up with detectable levels, and some don't change at all. Some of that relates back to brush design, rotation, and population size. So just there's a lot that has to go into a properly designed research project. No big surprise there. I'm going to shift a little bit again to back out to the field and leafy greens, just to because I started getting a lot of questions around this. Again, this is older data. Had to stop partway through for a variety of reasons I won't go to, but we're looking at trying to understand what would be the likelihood and what is the best methodology for recovering listeria from soil associated with crop, to ask the question. Yes, we know it's really nice studies that have come out since then that show obviously area, region, zone, the prevailing conditions really make a difference as to how you define prevalence. But pretty large field trials inoculated onto small red chard and then dished into the field. And what we're looking for, what we found, is by having this rifampicin antibiotic selective marker, if we didn't use it, 25-gram soil sample was non-detect at 32 days post-disking. 100 grams was 25% at the same point. So more sample, more likely to detect. And by 50 days without the rifampicin selective marker, we couldn't find it. Using that rifampicin marker, again, with enrichment and PCR, at 100 gram soil, 100% positive, still at 60 days. So it's hanging in there, but non-detected 77 days. So again, field trial, not reality. We took that sort of approach out to the field, and just briefly sharing a soil survey, 172 samples, typically 15 per field composited, sampled at soil moisture, disked 1 week. So this is a commercial crop prior to sample collection, all negative using the same techniques, pulling 2 water samples from adjacent to the field. There was a ditch with lettuce residue, happened to be negative within the sampling regime we used. The creek nearby was positive for Listeria species. We only look for Listeria species. So again, we couldn't continue because the cutting was dropped off. Once we found that result, we said, we're good. No need to go further. But it indicated that it can happen. It does happen at some level, but not likely. That was then used years later in a rapid response associated with asking the question of detectability of Listeria monocytogenes at a particular ranch location. I can't remember exactly how many sample cores were taken, but as I recall, it was more than 60 right along the edge, and then further in at each along a transect. Could not recover detectable levels of Listeria monocytogenes, even though one could readily obtain it from heavily impacted creek nearby. So again, the research can help drive then how you use that for meaningful response. Many of you have heard me talk a lot about biofertilizers and biostimulants as a real concern, a risk concern. I shared some examples the last time. I didn't want to really go back to that entirely, but again, this can be something used on the farm, within your water, or in a neighbor's farm, and it goes back into runoff impacting a shared and common canal system. Just again, touching back on what we've known for several years now, that fertigation with soluble biostimulant nutrients has led to very large scale contamination or contamination of water distribution system. And the products themselves are not necessarily contaminated, although I'm pretty strong evidence that in one case that was really happened at the distribution yard, but it's really coming from the makeup water. So again, manageable, preventable, just have to be aware. This one's a little bit different. Again, I'm focusing on this because of the pervasive and widespread marketing, development, and pushing biostimulant formulations for some very good values and benefits, but again, not a comparable attention to some of the manageable risks like salmonella, especially. Very competitive. Here, its biostimulant formulation was used to control or theoretically control tomato brown rugose fruit virus. It triggered a very large scale salmonella contamination in shade house production due to the presence of milk proteins and sugars and protein hydrolysates and a variety of proprietary additives. Once contaminated with makeup water, it grew, it persisted in the spray tanks, it persisted in the lines, and of course, it caused a serious crop destruction not associated with the virus. A conclusive source was elusive, but survival post-chlorination through the distribution system and then regrowth or growth within that formulation is plausible based on the data collected. I won't go into it, but the water source is a covered but basically open reservoir. Did go through a chlorination system, which fundamentally I think was very well designed, but I think there's some ways and means that need to be readdressed at how salmonella in particular managed to escape their sublethal injury, but not non-viability. Switching then to a little bit more, and Laura yell at me when they move me along or something like that. I may not make it. I know I'm talking a little bit excess here, but just the luck of timing. And there's no substitute, no way to plan for the serendipity that happens when you're out in the field, boots on the ground, and being observant has really led to some strong investigative testing and changes of practices, or at least suggestion that practices would change. What I'm showing here is that there is a very short and specific time of year when mud swallows are coming in to collect mud from the overspray. Orchard, I've observed this a couple of times. You come back any other time of year, you'd never see it, but there's bird droppings all over the farm roadway and water is going back and forth. And when they are going to cultivate, and you can see in the picture on the left, you move pipe, you move main line, there's chicken manure pellets there. There's a variety of ways for amplification to happen or to get into those sprinkler joints and potentially result in, particularly for a short crop cycle, some contamination that may occur. We've had multiple opportunities with pigs. I won't go into depth, but this is one where just early days of trying to establish buffer zones due to an intrusion event and tracking particularly where little piglets ran, and just from their external contact transmitted to a standing lettuce crop. I showed this one at the beginning. I just thought I'd share. It's not just lettuce and leafy greens, but Porky and friends were enjoying local melons. And it may sound weird, but one of the fun things is to get a call on an early Sunday morning and say, hey, can you come out and help, about 20 miles away from Davis. We've got pigs enjoying our melons and we don't know what to do. For some of us, that's a real rush. Anyway, moving on. Extension working with industry does play a key role in turning academic and translating academic research findings into rational actions, hopefully. Developing SOPs and science-based database metrics for both species-specific and situation-specific bird intrusion. Again, a number of things where it's the species, but the number and where they've been and where they're visiting and what else they're doing in and around the farm that really make a difference of risk to no risk, and ways to go out and sample and start testing to say, is this one of those situations where we have a problem or not? But I think that's really led to some future novel remote sensor activated bird diversion technologies that are species specific. So again, that all that happens. I'm going to try and go through this pretty quickly, hopefully make sure that there's time for questions or comments or whatever. So why is root cause assessment so hard? What is the fix? I'm not sure I have a great answer for that. More often than not, at least in my experience, there's truly a deficit of facts, incomplete information, and even misdirection or CYA. If you don't know what that is, cover your ass mentality. And it really is a barrier when you're there to try and help. Understandable, but not productive. And we really encourage, try to build a culture of trust, and trust is earned. Believe me, and there's many wonderful examples of people that have engaged in this same type of extension, outreach, applied research while balancing that with more fundamental or basic research. There's a great up and coming population and established population, certainly within this contact effort that does that. Build a sense of security that you understand their culture, their needs, their sensitivities and potential impacts on their business, on their region, on their commodity. But also, I would say, don't always give a second chance for failures. That's a little rough, but that comes from experience. I've learned over the years to really embrace this is and isn't approach to problem solving. You have to pay attention to what appears to be the is data as well as the isn't, and does it make sense? And develop a detailed matrix or checkboxes of where information lies into each one. And you got to be honest, you got to be fearless in attacking it. So I'm going to quickly run through an example, which I think is a fun one. It's not as scary as some others. Hopefully you'll stay with me. This is a grower's real life reality. This is part of a team. I will certainly acknowledge the tremendous team working with Taylor Farms, as in many firms, but really dedicated to come through this risk assessment and make tough decisions. So the harvest is planned minus day 6. Food safety team wins as a pre-harvest walk around. They do pre-harvest sampling on day 4. They pulled the drip tape on day 1. The harvest foreman sees cow pies in the corner of the field as this is going on. Hopefully you can follow me in all of this. Calls the farm manager, stops harvest staging, informs the food safety team leader all the things that are supposed to happen here. And the food safety team goes WTF. And they start to activate the risk assessment SOPs that they have established. And so the neighbor, the farm manager alerts the neighbor alerts the farm manager 3 days earlier that the herds got loose and they're back on the hillside, crop and physical grazing damage and multiple cow pies, but just in a corner of the field. The gate was open. The cattle took a pre-dawn field trip 3 days before planned harvest to the adjacent farm. And that all became part of the risk assessment. Oh, by the way, the farm manager says, oh, those were my cattle. Oops. So they get together and they come up with an assessment and action plan based on the observation of the cow pies. They establish a no harvest zone, all drip irrigation. 3 days from the last irrigation, there's no water pooling in the no cow pie zone. So maybe we can harvest that. Oops, wait a minute. There's fresh tractor tracks in the furrows were noted during the walkthrough. So they have to come up with a go, no go harvest decision. There was, by the way, 2 days prior to planned harvest, an insecticide ground rig spray. There was edible portion sampling and a pathogen screen. There was a spray pre-dawn to minimize drift. And the farm manager stopped that one. It didn't know that it was going on. It happened to pass through the cow pie zone in the first passes and ended up covering the full 12 acres. By the way, now the results come in with that kind of a sampling design. It was all non-detect decision time. Normally, this would be a challenge to the participants in the training session. And, of course, the decision is because of that tractor movement, we can't take a chance. It's all destroyed. So document the critical logic path, document all decisions, what you did and why, what you rejected or didn't do and why. Take a lot of digital images and then test to learn. Testing for that purpose to hopefully develop better preventive systems, better communications, better retraining are all important. So what is my point? Hopefully I feel that I tried to shoot for a point. There have been a few notable and reasonably transparent rapid response efforts, a number of them funded by CPS with industry. Knowledge is shared and adoption where results indicate. So my question is, why can't this be organized and regionally implemented? That's just an image of one of the multiple teams that I wish I could acknowledge, however many there were over the span of years, 40 or more, between staff and students. And so I originally proposed this sort of model rapid response and root cause assessment. I'm not going to go into great detail, but just to share two things. One, that you can decide on what the question is, what the opportunity is, foundational practical knowledge. You may be triggered by risk observation or site hazard assessment, but going to the grower, handler, or other to say, do we want to go through this? And then here's the complex piece, originally designed to pull in the different stakeholders to try and cut down the silos, really learn from this effort. Obviously, it's a bit complicated. It's way over the top from things that they used to do, but really was dictated by the concerns and the issues around. How do we do this on a regional basis? How do we support and fund this so that it is more available so we can take advantage of these opportunities when they come up? Again, I'm just not going to go into great detail, but I think there are ways that we know in the future we can ask, why can't this happen? A very much reduced version of this scheme was supported by Western Growers Association twice in response to requests for assistance due to either agency surveillance sampling positives or somebody else getting a positive. And it was all restricted up into this area, not bringing any others. And I'll just share with you that each time that was started, there were reasons that the industry decided to pull out. So it never went very far, but a number of things were learned along the way. And some of those go into that biostimulant category. For produce safety systems, this is just a bit of my own thoughts, a look to the future, that the best future depends on industry making better use of available science and what we already know, as well as what we can learn in the future, and the practical integration of knowledge, experience. And really this, in part, I think for the future, have to go further in developing the next generation of food safety researchers and food safety professionals, and especially encouraging the next generation of applied and mission-oriented research and extension specialists. And again, I've already acknowledged there's a lot of bright, shining stars out there. We just need a lot more, and we need them more distributed, and we need their institutions to recognize their efforts and their contributions. From the grumpy old guy who's yelling, "Get off the lawn," that's me. What are some of the barriers to professional development and cross-culture communications? I realize this is a bit blunt, but understand there's a reason that I wanted to share this. I think we have to overcome, again, there's some wonderful positive examples, but in general, work ethic problem or limitation that one experiences a lot. And I'm talking across a lot of different levels, and entitlement attitude that we see, and a degree status attitude. I'm not saying that didn't exist before, but I think all of these in this arena are something that we could work towards, and acceptance across the board for everybody, all stakeholders involved in coming together, breaking down those silos and working towards a common goal. Is you have to understand and accept that you don't know what you don't know. And if you're offended by any of these statements, then maybe it doesn't apply to you, and I'm not talking to you. And one of the things that stood out to me the last 2 years, the Center for Produce Safety had a postgraduate industry immersion program that I helped develop with Bonnie Fernandez-Fenaroli, designed to provide this unique research and musing experience built around focused research objective with an industry partner between the academic major advisor, the student, to really encourage people either to go into industry or to really focus on extension training, which is, I can say this just because I had that opportunity largely, and that's what I wanted, and I went that direction. It's really to support continuing professional development of promising master's postgraduate students, catch them at that stage, whether they go on for a PhD or they stop there. In 2 years, there's not been one inquiry, not one interest, even though many companies have offered to host that. So that's out there. And if you're interested, I think it's a great opportunity to do that. So then lastly, filling these knowledge and solution gaps, really emphasize breaking down silos, invest in understanding the cultures and motivations, both directions. It's important all the way around, particularly if you're going to try and understand how to improve the types of research that's asked for, the types of objectives, whether it's a USDA, SCRI, or a NIFA, and the panels that look at that. For the industry, share the diversity, make sure that the research community understands what you do, what some of the sensitivities are, and there's a improve communication around that as opposed to what seems to still be going on too many places today is something. And when I say build accountability systems, I'm also talking about, I think we need to build better accountability systems for where research funding is directed to improve the outcome and the adoption of that. So I'm going to stop with that.
Thank you. Thank you. And we'll see everybody back June 27 for Ag Water.